Very good evening to all the students. So, today we are going to discuss on shorthand type of questions from photosynthesis in higher plants and respiration in plants. As we have dealt the clear differentiation between the double membrane bound organelles, that is chloroplast and mitochondria, and also we have discussed what chloroplast structure in cell biology previously. Now, we are going to start from C4 pathway directly. If you want, we will go for this also. So, if you see here, draw a neat label diagram of chloroplast. This question is there in cell bio as well and in photosynthesis as well, okay, because in double membrane bound organelles of cell, the unit of life, we have this question that is about chloroplast, and multiple times asked question, like in 2024 might be, in 2023 might be. First, what you have to do is, beta, whenever diagram based question you are attending, first draw out the diagram, then that diagram stands as a reference to explain or elaborate the theory below. First draw the diagram and then go for the theoretical explanation of the answer. So, here if you see, it is double membrane bound organelle, it has two membranes, outer membrane and inner membrane. Outer membrane and inner membrane has periplastidial space in between. Okay, what does it have? Periplastidial space. And inner matrix you go through. First you explain about outer membrane, inner membrane, periplastidial space which is in between outer and inner membrane, and then you explain on the matrix of the chloroplast. Matrix of the chloroplast is stroma, and it has sac-like structures, membranous sac-like structures called as thylakoids. Okay, this is said as third membrane system of the chloroplast. Stalk of this sac-like structures is known as grana. One grana is connected to another grana by stroma lamellae. One grana is connected to other grana by stroma lamellae. Fine, right? Now you see, inside the matrix of the chloroplast you have granules also, starch granules and lipid granules. Fine, right? So here if you see, chloroplasts are the green double membrane bound organelles. These are found in the mesophyll cells of the green plant leaves. They are the main sites of photosynthesis. If you see shape and number of chloroplasts per mesophyll cell, chloroplasts are usually lens shaped, oval, discoid and sometimes ribbon like. Their number varies from one cell to the other. In Chlamydomonas, if you see, there is only one chloroplast per cell. In thylakoid mesophyll cell, if you see, we have 20 to 40 chloroplasts are present per cell. Structure, outer membrane and inner membrane, and elaborate about periplastidial space, followed by stroma, which is the matrix of the chloroplast. It consists of enzymes, DNA, ribosomes and starch granules. The dark reactions, those are Kelvin cycle of photosynthesis. This will occur inside the stroma itself, and thylakoids, if you see, these are flattened membrane bound sac like structures. These are present in the stalks called as grana. The thylakoid membranes contain chlorophyll and other pigments that capture light energy. The light dependent reactions of photosynthesis will occur here. Next, grana and stroma lamellae. The granum is a stalk of thylakoid and the grana are connected by what? Stroma lamellae in between. These are intergranal thylakoids that will help in the transfer of materials and energy. Other components, if you see, other components we have ribosomes. Those ribosomes are 70S ribosomes. 70S ribosomes whose larger subunit will be 50S and smaller subunit will be 30S. They are binded by magnesium ions. 70S ribosomes whose larger subunit and smaller subunit. Larger will be 50S, smaller will be 30S. Followed by functions of the chloroplast. Conversion of light energy. In the thylakoids, light energy is converted to chemical energy. This chemical energy can be ATP and NADPH. This happens during light reaction. Followed by synthesis of carbohydrates where the enzymes in stroma will utilize ATP and NADPH. This will synthesize carbohydrates during dark reaction. Till here it is clear, right? This is the complete structural and functional description of chloroplast with the diagram. Now we have to tabulate. Next question is on tabulate any 8 differences between C3 and C4 plants or cycles. Fine, right? Between C3 cycle and C4 cycle we have to tabulate minimum 8 differences. Multiple times asked question. If you see in 2023, 2018, 2017 and 2015 as well. These many times asked question. C3 plants, if you see, the first stable product of carbon dioxide fixation is 3 carbon compound, that is 3-phosphoglyceric acid. C4 plants, first stable product of carbon dioxide fixation, which is 4 carbon compound, that is oxaloacetic acid. Here if you see carbon dioxide fixation and Kelvin cycle, both of them, that is Kelvin cycle or carbon dioxide fixation, this will occur in mesophyll cells, whereas here 2 times carboxylation will happen, primary carboxylation and secondary carboxylation. First time the carbon dioxide fixation happens inside the mesophyll cells and Kelvin cycle occurs in bundle sheath cells. The key enzyme for carbon dioxide fixation is rubisco, ribulose 1,5-bisphosphate carboxylase oxygenase. And here in C4 plants, the initial carbon dioxide acceptor enzyme is PEP carboxylase and the Kelvin cycle will involve rubisco here. Here in C3 cycle, these plants will undergo photorespiration. This leads to loss of carbon dioxide and energy. C4 plants, photorespiration is completely absent or negligible. This is because carbon dioxide concentration in bundle sheath cells will be high. Just if you want, you can take a screenshot as well, but this is how you have to explain crystal clear differences between C3 cycle and C4 cycle. Fine, right ma? Now, yeah, here we go. They are less efficient on hot, dry climates due to photorespiration. Fine, right? They are less efficient due to what? Photorespiration. And if you see in C4 plants, it is more efficient in hot and dry climates. This is due to carbon dioxide mechanisms. Okay, that is due to carbon dioxide mechanisms over here. Fine, right? Yeah, here we go. This is for C3 plants and C4. Here chloroplasts are similar in all mesophyll cells, whereas here chloroplasts are dimorphic, granule in mesophyll cells and agranule in bundle sheath cells. Here you see carbon dioxide fixation occurs only once through Kelvin cycle. Here carbon fixation occurs twice. This will occur twice. First by PEP carboxylase in mesophyll cells, second by rubisco in bundle sheath cells. Fine, right? Next. Here examples: wheat, rice, oats, barley and sunflower, and in C4 plants maize, sugarcane, sorghum, millets. These will be the examples for C4 plant. If you can take a screenshot, it will be clear for your reference. Very important, ma. And then we will go on to the question which is very important, that is describe C4 pathway. Okay, appeared in 2019 and 2016. First, I will always recommend to have a schematic representation like this. First to draw the basic diagram and then start on elaborating the concept. I will explain you here itself and then we will go with the theory. These cells are what? Mesophyll cells and bundle sheath cells. How they are connected? Mesophyll cells and bundle sheath cells are connected by plasmodesmatal connections in between. Fine, right? They are connected by plasmodesmatal connections. Here if you observe, carbon dioxide is fixed into the mesophyll cells in the form of hydrogen carbonate. Okay, this is fixed in the form of hydrogen carbonate. Once it is fixed, that C4 acid in the form of oxaloacetic acid, this is being transported to bundle sheath cells. In bundle sheath cells, this 4 carbon compound will undergo decarboxylation by releasing out one carbon dioxide and C3 acid. This carbon dioxide will undergo C3 cycle, that is Kelvin cycle inside the bundle sheath cells, because bundle sheath cell is rich in rubisco. Rich in rubisco. Fine, right? And this is transported, the C3 acid is transported and regeneration occurs. Okay, it is regenerated back in the form of pyruvate. Fine, right? Inside the mesophyll cells. Clear, right? Now we will go on elaborating the concept. So if you see the theory of this beta, the C4 pathway, also known as Hatch and Slack pathway, which is named after the scientists Hatch and Slack, is carbon fixation mechanism. This is found in certain tropical and subtropical plants like maize, sugarcane and sorghum. It is an adaptation to minimize what? Photorespiration, and this will increase photosynthetic efficiency even in the dry climates. This is the adaptation of what? C4 plants. This occurs in 2 types of cells. One is mesophyll cells and second will be bundle sheath cells. Mesophyll cells, initial carbon dioxide fixation will take place. Bundle sheath cells, where Kelvin cycle will operate. This special leaf anatomy is called as what? Kranz anatomy, where bundle sheath cells will form a ring around the vascular bundle. Steps which are involved in C4 pathway. First, carbon dioxide fixation in mesophyll cells, where carbon dioxide enters the mesophyll cell. This will combine with PEP, phosphoenol pyruvate, to form a 4 carbon compound, that is oxaloacetic acid. This reaction is catalyzed by what enzyme? PEP carboxylase enzyme. Okay, this is catalyzed by, the first step is catalyzed by PEP carboxylase. And then formation of malic acid or aspartic acid. Here OAA, that is oxaloacetic acid, is converted to malic acid. Both are 4 carbon only, which is an isomerization reaction. These acids are transported from mesophyll cell to bundle sheath cells via plasmodesmatal connections. Next, decarboxylation in bundle sheath cells. Inside bundle sheath cells, malic acid is decarboxylated to release carbon dioxide and a 3 carbon compound, that is pyruvic acid. The released carbon dioxide will enter the Kelvin cycle. It will enter Kelvin cycle, where it is fixed again by what? Rubisco. This is fixed again by rubisco to form sugars. Thus, carbon dioxide is fixed twice, once by PEP carboxylase and second time by rubisco inside the bundle sheath cells. So, here malic acid is converted to carbon dioxide and pyruvic acid. Now, regeneration of PEP. The pyruvic acid returns to mesophyll cells, where it is converted back to phosphoenol pyruvate using what? ATP. This will use ATP. This regeneration will ensure continuity of the cycle. This regeneration will ensure continuation of the cycle. Fine, right? So, first you make this diagram clear and then elaborate this properly step by step, where first carboxylation is happening, where second carboxylation is happening, what are the enzymes involved there, everything you have to mention. Highlight Cran's anatomy as well. Now the energy requirement and significance. Most important, right, bioenergetics, right ma? The C4 pathway will require how much ATP? 30 ATP to produce one molecule of glucose. Despite this, it is more efficient because photorespiration or C2 cycle is absent. Okay. So here photorespiration or C2 cycle is absent, followed by significance of C4 pathway. This prevents photorespiration, conserving energy. This increases carbon dioxide concentration around rubisco. This will enhance carboxylation efficiency. This will enhance carboxylation efficiency. Highly efficient under high light intensity and temperature. Fine, right? Next, this reduces water loss, and this is due to partially closed stomata. Examples you see, we have maize, sugarcane, sorghum, millets and amaranthus. These are the examples for C4 plants. Now, explain briefly about photorespiration or C2 cycle, important. C3, C4, CAM and C2 cycle, all are important beta. So here photorespiration, this is again a light dependent process. C2 cycle operates only in C3 plants, remember, because C4 plants have an adaptation where photorespiration is not functional in C4 plants. Photorespiration is done only in C3 plants. Okay, C2 cycle is operated only in C3 plants, where the enzyme rubisco binds to oxygen. When oxygen levels are high around the atmosphere, then what happens? Rubisco, as it has both oxygenase and carboxylase activity, rubisco will bind to what? Oxygen instead of carbon dioxide. This leads to oxidation of RUBP. Fine, right? This leads to what? Oxidation of RUBP, ribulose bisphosphate, and release of carbon dioxide. This process does not produce ATP or sugars, and hence it is considered as what? Wasteful process. Consequence of photorespiration. What is the consequence of photorespiration? This wastes energy, no sugar or ATP is produced, rather energy is consumed and carbon dioxide is released. Fine, right? This leads to carbon dioxide loss instead of fixation. This increases at high temperature, reducing crop productivity. And these are the organelles which are involved in photorespiration. Complete mechanism you have to highlight. Okay, just look into this. We will get back in a minute. Here RUBP binds with oxygen. Oxygenase activity will happen where 5 carbon RUBP will give out two products. One is 3 carbon phosphoglyceric acid and 2 carbon phosphoglycolate or phosphoglycolic acid. Here 1 phosphoglyceric acid will not continue with C3 cycle, right? It requires 2 PGA. To synthesize one more PGA, a whole lot of process is operated, that we are learning it as what? C2 cycle, where it travels across 3 organelles, that is chloroplast, peroxisome and mitochondria. utilizes energy, releases of carbon dioxide. As such, it is considered as wasteful process. So here phosphoglycolate is first converted to what? Glycolic acid or glycolate. This glycolate is again converted to glycine in the peroxisome. Glycine is converted to serine in mitochondria. Here itself carbon dioxide is released in the mitochondria and NADH is also released. Serine enters the peroxisome, converts to hydroxypyruvic acid where NADH is utilized in this process and it is releasing glyceric acid or glycerate. Glycerate enters chloroplast, undergoes phosphorylation where ATP will give up that phosphate group and phosphoglyceric acid is formed. So here if you can see, energy is utilized, carbon dioxide is released. There is no amount of energy that is released in this process. Of course, NADH which is released in the mitochondria, in the entire process again it is utilized in the peroxisome. So this is neutralized. On whole, ATP is utilized and carbon dioxide is released in the mitochondria. Till here it is fine, ma? Yeah. So this you have to draw out primarily in the beginning of the answer and completely you explain this in detail, the complete mechanism of action of C2 cycle. Then move on to site of photorespiration. Photorespiration usually occurs in chloroplast, mitochondria and peroxisomes. So this is the complete process. Just go through the entire process. If you are not getting anywhere, just ask me. What all we have discussed over here only, it is there here. Just go through it and ask if you are not getting anywhere. And now here we move on to next chapter, that is respiration in plants. First question, describe briefly the process of fermentation. Here you have to explain two processes of fermentation. One is alcoholic fermentation, second will be lactic acid fermentation. So here fermentation is what? A type of anaerobic respiration in which carbohydrates are partially oxidized or incompletely oxidized to produce organic acids, alcohols or gases such as carbon dioxide. It occurs in microorganisms like yeast and bacteria and even in muscle cells of animals during oxygen deficiency. Fine, right? So now here you see what is the process of fermentation. Anaerobic breakdown of glucose. Here fermentation takes place under anaerobic conditions and glucose is only partially oxidized to form what? Pyruvic acid. This is through glycolysis. Now conversion of pyruvic acid, this takes place in two pathways. One is alcoholic fermentation, that is by yeast. In yeast, pyruvic acid is converted to ethanol and carbon dioxide. It occurs with the help of two enzymes. One is pyruvate decarboxylase, second will be alcohol dehydrogenase. Next, lactic acid fermentation. This will be by bacteria and this will occur in bacteria like lactic acid bacteria, and this also occurs in muscle cells or muscle tissues of animals. In bacteria and oxygen deficient muscle cells, usually in the time of vigorous exercise where oxygen is supplemented by lactic acid fermentation in the muscle cells. That is the reason at vigorous workout people will get muscle cramps because of lactic acid accumulation in the muscle cells. Here pyruvic acid is converted to what? Lactic acid. This is catalyzed by the enzyme lactate dehydrogenase. Pyruvic acid is converted to what? Lactic acid. Energy released here is fermentation will release very little energy compared to aerobic respiration. Only 2 ATP molecules are produced per 1 glucose. And here types of fermentation, if you see, alcoholic fermentation and lactic acid fermentation. Alcoholic fermentation, ethanol and carbon dioxide. Example you can see is yeast, Saccharomyces cerevisiae. Lactic acid, here example you can take as lactobacillus and human muscle cells. Energy yield, net gain is 2 ATP and fermentation is thus low energy yielding process compared to aerobic respiration, which yields around 36 to 38 ATP. Now, next question is about RQ, that is respiratory quotient, which is asked in multiple years like 2022, 2018 and 2016. Here respiratory quotient is the ratio of what? Volume of carbon dioxide evolved by volume of oxygen consumed. That is the standard formula of RQ. Here you have to give out RQ of carbohydrates, fats and proteins. First we will see RQ of carbohydrates. Here if you see carbohydrates, for example glucose, they undergo complete oxidation. The volume of carbon dioxide evolved equals to the volume of oxygen consumed here. Okay, volume of carbon dioxide evolved by volume of oxygen consumed. Give this out with the formula. Okay, glucose plus oxygen at the time of complete oxidation will give 6 carbon dioxide, water and energy. Here how many carbon dioxides are evolved? 6. 6 carbon dioxides by how many oxygens consumed? 6. It is equal to 1. So here RQ value is equal to 1 and there are no units over here. RQ for fats. Fats contain fewer oxygen atoms, more carbon and hydrogen atoms compared to what? Carbohydrates. So therefore more oxygen is required for what? Oxidation. More oxygen is required for oxidation here. So here volume of oxygen evolved will be always less than making up what? Oxygen consumed. So here oxygen will be more than that of carbon dioxide evolved. So in denominator more number, numerator less number means the value will be less than 1. As such RQ value for lipids is approximately 0.7. Now we will see RQ value for proteins and organic acids. Yeah, with this we can end 2 chapters, photosynthesis and respiration. Here we go with RQ value of proteins. Yeah, when proteins are used as respiratory substrates, the amount of carbon dioxide evolved is slightly less than that of oxygen consumed. Hence, RQ value will be equal to 0.8 to 0.9. Now, RQ value of organic acids. In this case, organic acids which contain what? More oxygen, the carbon dioxide is evolved more than the oxygen consumed. Here CO2 evolved is more than the oxygen consumed. Therefore, RQ value will be always greater than 1 for organic acids. Malic acid plus 3O2 gives rise to carbon dioxide and 3H2O. Here 4 by 3, which is the value is more than 1, that is 1.33. Fine, right? So, with this, the short answer type of questions for photosynthesis and respiration are done. And to access the soft copies of the complete materials provided by NEET World, it is very simple. Follow us on the Telegram and Instagram and just say hi in our app, that is MedWorld. Install it from Play Store, wherein you will get all the 4 soft copies of your IPE material, which are well structured, crafted, and in botany the long answer type of questions are given with flow charts and mnemonics, which will be very useful for all the students. So, get it accessed as it is completely free of cost. So, with this I will end up today's session. We will meet in the next class. Thank you.